All right, welcome to Chapter 5, Part 2. We are going to finish up Chapter 5, and this is kind of the fun part of viruses, if you want to think of it that way. Lots of different things to talk about. First, we have the three types. You can kind of think of these as forms or kinds of infections that you can get. The first is acute. Does not mean it's adorable, it means that it's quick. We are talking about it is quickly coming on as far as an infection goes, and it is going to leave quickly. So examples include like the cold and the flu. Another type of viral infection is a latent viral infection, sometimes called chronic. This is going to be an infection that remains asymptomatic, so no symptoms in the host for certain periods of time. This is going to be a dormant period, so inactive, but that doesn't mean that the virus is gone. It just means that the virus is not active at that moment, causing symptoms. It can periodically emerge where there will be symptoms, and that can be caused by various things. So some examples: cold sores, herpes, shingles. Persistent viral infections are going to be where the individual infected is going to have symptoms the entire time for the infection, and typically these are going to result in fatal conclusions. So you can think of things like HIV as being a decent example of a persistent viral infection. So you're going to have an active virus, so there's no dormant periods, and the individual is going to be showing symptoms for the duration of that infection until potentially and unfortunately death. So looking at a graph of our types of infections and then the number of virons at those times, so the number of that virus, we have that acute infection. Again, it comes on quickly and it also leaves quickly. So those numbers of viruses in the host at that time is going to be very quick, and then it drops very quickly as well. For a latent infection, we're going to see kind of a gradual rise, but it's not going to be a particularly large amount of virons. And this is again going to be during an outbreak of some kind. And then you have a persistent infection where it's going to be the symptoms are always present, the virons are always present, and you have kind of a ramping up event where the number of virons are going to increase, the symptoms are going to increase, potentially get worse until you get to this point where potentially death is going to occur, wherein the number of virons and the infection then end. Now, we talked about the different kinds of infections. We do want to talk about the different kinds of viruses. So there are 4 kinds of viruses that I want you to know. Here are the first 2. We have a provirus, which is going to be the type of virus that is going to infect animal cells. Very important. These are going to be the types of viruses that can infect us. There are virophages, which are going to sound really neat. But in the end, it's a little bit scary thinking about it. These are viruses that can infect other viruses. Now, this sounds great, like woohoo, we've got an inside man. We can take out the viruses from the inside. Well, it's not really something that we have been able to successfully use to attack viruses. Otherwise, you probably would have heard about it up until now. And we've only recently discovered that this was something that could happen in 2008. So if you want to think of it like this, if there's a madman, he's taking control of a train. That's kind of like that original virus. And then someone else comes along, takes him out. Well, now you just have a different matter man taking control of the train. And that's what happens with virophages. You have the original provirus potentially infecting the host cell, a virophage can come along and take over that provirus, and the virophage just takes over the job of the provirus from before that it is now infected. So it's not that it is removing the virus from the host cell, it's just taking the place of it. The third type of virus is an oncogenic virus or an oncovirus. These are viruses that are going to be linked to cancer. So it is estimated, and this is a pretty high estimation, that 20% of human cancers are caused by viruses. What it does is it causes the host cell to transform and has all these different changes, increased growth rate, alteration in the chromosomes, changes in the cell's surface molecules, and the capacity to divide indefinitely, which is all definitions of cancerous cells. So some examples: HPV, hepatitis in the liver, which can cause liver cancer. So remember we were talking about at the end of chapter 5, part 1, that when it comes to viruses leaving a host cell and it survives, not the virus, but the host cell, it can result in that damage to the host cell, even if the host cell survives the virus itself. This is going to kind of play into this oncogenic virus as well. So remember, it can cause the host cells that survive the infection to become sticky, multinucleated, and kind of grow without inhibition. That is cancer. Oncogenic viruses can contain something called oncogenes, and these are naturally occurring DNA sequences that we also have in our cells. So in our genes, we also have oncogenes, except a little bit of a difference. For viruses, these induce cancer. For our DNA, our genes, these oncogenes are going to regulate cell growth and prevent tumors. What viral oncogenes do is kind of pervert that process. And the other thing that's important to note is that the viral oncogenes are stronger than ours and will take over the process of cell growth within a host cell. And it's kind of almost like zombie mind control, where it causes the cell to just grow indefinitely and causes those cancerous buildup of cells. Here are some examples of oncogenic viruses. You do not ever need to know examples unless I tell you to. This is just to help you kind of visualize what we're talking about here. So we have some examples in DNA. We have Epstein-Barr virus. This is going to be a DNA virus that causes cancer, as well as a handful of other ones you've probably heard of, the human papillomavirus or HPV. This can cause cervical cancer. There are also RNA oncogenic viruses, and all of them are retroviruses. We talked about one of these before, and that is feline leukemia. This is a very scary condition to have if you have cats, because it is an infectious virus that causes cancer in cats. So super, super bad time if you have kitties. Naturally, our last type of virus is our good friend, the bacteriophage. We've talked about him a few times up till now, and that is our fourth type of virus. It is going to be specifically viruses that are going to infect bacterial cells. In fact, the term bacteriophage means bacteria eating. Now, because bacteriophages are just so gosh darn neat, we are going to talk a little bit more about them. They are more complicated than other viruses, and we've actually kind of talked about what happens in one of their cycles, their reproductive cycles already. They do have two distinct cycles, though, the lytic cycle and the lysogenic cycle. The lytic cycle is going to be that viral life cycle we discussed in Chapter 5, Part 1 already, so those 6 steps from before. So we did talk about that before, and we are going to review it on the next slide, but let's talk about that lysogenic cycle. So this is going to be where the virus itself is going to hide in the DNA of the bacteria, because remember, the bacteriophage is going to inject its nucleic acid into the host cell, into the bacteria. And that little bit of viral nucleic acid is going to hide in the DNA of the bacteria. And then as the bacteria does its natural process of binary fission, dividing, it spreads the virus without realizing it. So it's not going to attack the virus DNA or RNA that is inside of its own DNA. It is just going to keep replicating that nucleic acid and spreading it to those new cells. That is the sneakiness of a bacteriophage using that lysogenic cycle. So again, it's going to hide in a single chromosome. The bacteria is just going to keep doing what it's doing, which is to replicate. And as it replicates, it is replicating that virus. So it's doing all the work. And at some point, the virus will eventually jump ship as the host bacteria begins to die. So let's go through these two processes a little bit more. We'll review those 6 steps and then talk about the lysogenic cycle a little bit more in depth. So we have these 6 steps minus 1. So remember that there is no uncoating phase for bacteriophages. So technically the 6 steps from before, it's technically 5 when it comes to the bacteriophage. I'd kind of make sure you know that. As far as the lytic cycle, it's just those 5 steps. Again, minus the uncoating, because the bacteriophage with the penetration is going to inject the nucleic acid into the bacterial cell. There is no need to take off that protein coat on the outside of the nucleic acid because it doesn't have one, because it's already penetrated the cell. The nucleic acid is already free of that coating. So the main thing here for the lytic cycle is get in, make new viruses, get out. That's the big thing. So it's still those same steps from before, minus the uncoating, get in, make new viruses, get out. For the lysogenic cycle, we kind of want to compare this to the lytic cycle. It's a little bit different. It's still kind of the same. The first 2 steps are the same as the lytic cycle, but then we have a few little differences. So there's a lot of kind of sitting and waiting after the virus is attaching and embedding itself into the DNA of the bacteria. From there, the bacteria is going to replicate its DNA and make a new cell. In that process, it is making a new virus nucleic acid that is going to be in the new cell that it has created. It's going to keep doing this over and over. What's kind of crazy and weird is in a lab setting, scientists wanted to see how long a bacteriophage would stay in this lysogenic cycle, and the longest in a lab setting was 12 years. So what is the end of the lysogenic cycle? Well, that is the excision. The excision stage of the lysogenic cycle is going to be where the bacteriophage recognizes somehow that the host cell is dying. So the bacterial cell that it is riding along with is dying. Much like rats on a sinking ship, the bacteriophage will jump ship. Okay, so how they do this is that DNA or RNA is going to remove itself from the bacteria's DNA, make those bacteriophages, and then jump out of the bacteria. So that is that lysogenic cycle. That excision stage is kind of the weird one. Again, the host cell is dying, and the bacteriophage recognizes this, ends up making those bacteriophages with some of the DNA that it has replicated, and then jumps ship. So this image is going to be the lytic and lysogenic cycles of bacteriophages. You don't have to memorize all of this imagery. It's more so just for your viewing pleasure. Help you understand those different processes. Sometimes imagery is the best way to make sure you understand it. So when it comes to bacteriophages, why the heck do we care? These are viruses that affect bacteria. What difference does it make to us? Well, when it comes to these bacteriophages, what they provide to the bacteria host cell can sometimes be a little bit of like an extra ability or superpower due to that combining or addition of the DNA of the virus to the bacteria's DNA. So kind of crazy to think about because remember that bacteria are going to have its own DNA, and remember that DNA makes us, right? The virus is changing that DNA. It is adding something to the DNA of the bacteria. So it's going to add this ability, usually in the form of a new toxin, to bacteria. Now, this doesn't always happen. This is not an every single time process, but it can happen. Some examples, and again, you don't have to memorize the examples, but some examples you can think of are diphtheria. This is going to be a bacteria that can cause a coating on the back of your throat, and if it covers the entire airway, it can suffocate you and kill you. Another is cholera. Certain strains of cholera, if they have a particular toxin that is strong enough, it can cause essentially super diarrhea, where you die from dehydration because you are having such severe diarrhea, you cannot hold any liquid in your body whatsoever. Water is not an option. You just, it's all gone, and you die of dehydration. Very scary, and it's because of that toxin being released from the bacteria that was added due to a bacteriophage. The last is botulinum toxin in Clostridium botulinum. It is another bacteria that was infected by a bacteriophage, releases this toxin that can paralyze your muscles. The big concern here is if the diaphragm, the big muscle that allows you and helps you breathe, becomes paralyzed, well, then you can't breathe and you will die. So this is the big scary concern of bacteriophages. While they don't always immediately kill their host, as far as in that lysogenic cycle, they don't kill their host immediately. They just kind of ride along getting replicated. That's fine. That DNA, or I'm sorry, that bacterial host cell is surviving, so it's not killing the host cell, which is abnormal for bacteria, right? It can become a real big problem for us as a side effect of that toxin due to that new DNA being added from the virus to the bacteria. Crazy. So gentle reminder of that process of transduction. Remember, that is that horizontal gene transfer is going to be happening because of bacteriophages. Remember that bacterial DNA is being transferred from a donor to a recipient cell by a bacteriophage, and this is another way that bacteria can develop a new toxin, which can be kind of scary. This is that handing over, if you want to think of it, of new abilities. I do want to preface this with, in that lysogenic cycle, it can usually lead to mistakes in the bacteria. So it's not always every single time that if a bacterial cell is infected with this lysogenic cycle of bacteriophages, immediately they get a superpower. That is not the case, not every single time. In fact, most of the time it leads to a mistake in the bacteria, and then that becomes an issue for the bacteria more often than it becomes an issue for us through the bacteria. Okay, moseying right along into cultivating viruses. What that means is we are growing a virus. Now, we will not be doing this in lab because it is entirely too dangerous, but other labs do this because they are able to, of course, makes sense. So I want to give you two terms here, and that is how viruses can be grown. So it's going to be like, think of it like this: location, location, location. Where is the virus being grown? You can have in vivo, which is going to be in a living organism, or in vitro. This is going to be growing a virus outside of a living organism. So you could be growing this in a cell or a tissue culture or a petri dish. It just depends. Now, I'm sure it's not too difficult to imagine what the purpose of growing a virus might be, but why do we grow viruses? A lot of research over vaccines, that's a big one, huge one right there. Being able to isolate and identify viruses, to classify viruses, maybe classify parts of viruses, discovering new methods for treating someone who's infected with a virus, etc. The list goes on. It's very important to understand viruses because we're constantly finding more, or they're mutating or evolving, if you want to think of it that way, into new strains. It's important to be able to grow viruses so that we can study them and therefore be able to fight them better and find better methods to treating viruses when we get infected. So how we grow viruses is going to depend very importantly on what kind of virus it is that you need to grow. So growing something like a bacteriophage, easy peasy lemon squeezy. Makes sense. You would use a petri dish or some type of media to grow bacteria and then have the bacteriophages infect that bacteria on a plate or a broth or whatever it may be. This is a decently easy virus to be able to grow, or a type of virus to grow. Remember that the first virus, quote unquote first, virus to be discovered was using plants. So we do know that plants can get viruses, and we want to be able to cultivate viruses that grow in plant hosts. And you can do this two ways: in vivo or in vitro. In vivo would be using the whole plant, but this is kind of expensive and it can take up a lot of space and it's time consuming. And if you're terrible at taking care of plants like I am, well, that might be a little bit more difficult. You also need all of the equipment to grow those plants, right? So growth lights, water, soil, all kinds of things. However, if you grow the virus in cell cultures, it's a lot cheaper, a lot faster, and a lot less time and space consuming. Provirus, or animal viruses, can be grown in 3 different ways. The first is in living animals, which sounds really inhumane, infecting a living animal such as a mouse, a rat, a hamster, guinea pig, rabbits, non-human primates, and occasionally insects with a virus. That is an option. Or growing a virus in animal tissue cultures, not dissimilar to that in vitro option with plants. And then that middle option there, you can also grow an animal virus in an embryonated egg, such as of chickens, ducks, and turkeys. The ducks of turkeys, not ducks and turkeys. Apologies. So when it comes to different vaccines of like the flu vaccine, things like that, we do cultivate this typically using eggs. So in that process of getting a flu vaccine, what you might be asked is, are you allergic to eggs? And if you are, they may recommend that you not take the flu vaccine due to a concern of a reaction, because there may be trace amounts of that egg in the vaccine where the virus had been grown. Maybe it was an inactive form of the virus. It just depends what we're talking about, but that is the reason why. Okay, switching gears a little bit to talking about particles that are infectious but not viruses. They're virus-like, or they act like a virus. Our first is going to be prions, and these are going to be very scary non-cellular infectious agents because of what they are, and we'll talk about that in just a second. What prions can cause is spongiform encephalitis. So what this is essentially a brain-eating disease that is caused by proteins. There can be a long period of latency, so no symptoms, and then suddenly symptoms come on. And these symptoms can come in the form of loss of muscle control, irregular speech patterns, a lot of stuff having to do with a problem with your brain, because it is your brain that is being affected. So what this protein does is it eats away at your brain tissue, making it kind of like Swiss cheese. It's full of holes. It is very sad, but this disease is progressive. It progressively gets worse, and it is fatal. It is absolutely fatal. There is no ifs, ands, or buts. Once you are infected with these prions, you have a life expectancy of 1 to 3 years. And 3 years is not necessarily a mercy in this sense, because you are essentially living through your brain deteriorating and there's nothing you can do about it. Which is very unfortunate. What's wild is we don't have a way to kill prions. And in fact, the only way that we know how to diagnose that someone is infected with prions is to take a sample of their brain. And that's very hard to do and still keep that person alive, based on where you need to get a sample from. So kind of spooky. Let's talk a little bit more about it. So as I said, prions are proteins. They are classified as infectious proteins. This is crazy to think about. Viruses, at the very least, are a nucleic acid, so some type of DNA and RNA, and a capsid. They're not a cell, but they're at least made up of DNA and RNA, but this isn't even that. It's just a protein, a naughty protein. And how they reproduce is even crazier, just by touch. So this prion, this infectious protein, just touches a non-infectious protein, just a normal protein, and it causes that protein to break. And in that breaking, it causes that unbroken protein to become a prion. And then that new prion becomes an infectious protein. And then it can keep doing what that original prion was doing, infecting other proteins. And that is how it works. Super scary, spooky, spooky stuff. But the nice thing, guys, is that this is terrifying, but it's rare. It's very, very rare. And we have a lot of regulation around how prions can be inherited and uh, uh, where you can get this type of infection. Okay. The biggest area is going to be ingestion. And unless you guys are eating copious amounts of brain tissue on the weekends, you have very little to worry about. However, when it comes to maybe the butchering process, I wouldn't look too far into it. Just know that it is not a clean process, that brain tissue and brain matter can accidentally get on meat that is not brain tissue. And if that happens, then what can occur is those infected proteins can get into beef products, and that is where we get mad cow disease. That is not to say that every cow that exists that is going to be slaughtered and then butchered and put out onto the shelves for you to make tacos on Tuesday, that is infected with prions. There are outbreaks of mad cow disease, and typically it is caught very quickly, very early, and then it is taken care of very quickly and early. Okay. There are certain areas that have been known for a prion outbreak, and there are certain diseases that are specific to those areas, such as Kuru. This is a tribe in Papua New Guinea that consumes raw organs and specifically brains of humans. It is part of a spiritual experience due to their tribes and warring. And what has happened in the past is that these infectious proteins have been consumed, and it causes a disease called Kuru. It is specific to this particular tribe, also known as the Laughing Death. But that is just prions, which is kind of scary. From there, another type of non-cellular infectious agent is a satellite virus. You can think of these as weak viruses, but they're not quite a virus. These need to piggyback onto an actual virus in order to infect a host cell. So the example that I gave you many lectures ago is what it feels like, is Borna agent being linked to schizophrenia. The connection that we're seeing is that it might be a satellite virus that piggybacks on another virus, infects somebody, and this satellite virus is what causes the schizophrenia after the original virus. So still a lot of research into these guys, but this is another one of those virus-like, not a cell, but kind of like a virus, infectious particles. Next up is a viroid, not to be confused with a viron. So make sure you know the difference between these terms. A viroid is going to be an infectious RNA molecule. That's it. That's all it is. It is virus-like, but all it is RNA, no capsid. What these guys can do is attack plants. They parasitize plants and can wipe out an entire agricultural enterprise, which is kind of terrifying. Imagine absolutely no potatoes whatsoever at all. I don't know about you, I really like potatoes. That would be a real big old bummer, as opposed to, say, chrysanthemums. While if those are your favorite flower, groovy, fantastic, that would be a real drag if you did not have chrysanthemums anymore, but not as critical as, say, not having entire food groups anymore, like tomatoes, potatoes, cucumbers. I believe there was some type of something similar, maybe to a viroid or an actual virus that attacked plants that took out a lot of bananas, or maybe it was a mold. Either way, it doesn't really matter. This does happen, guys. It's super scary. Very, very scary. But again, viroids are just RNA molecules that are infectious, not a virus, but it's a virus-like thing. And again, don't get these confused with a viron. A viron is a full viral particle. So contains a nucleic acid and a capsid. These guys don't have a capsid. They're just RNA. All right, on to what you probably know when it comes to viruses, and that is how it affects human health, and a lot of examples of viruses, ones you've probably heard of, maybe some you haven't. All right, so we have a nice laundry list of different viruses here. And the thing that I really want to get across is that there's a lot of different viruses that occur worldwide, and it's estimated to be in the billions. The thing with these examples is that they come in a lot of different flavors as far as being really common, aren't so terrible, like the common cold. Not a big deal. The flu, super sucks to get the flu, but it's not really like the end of the world. Same with warts. Is it not great to look at? Sure. Yeah. Are they bothersome? Yes, absolutely. But they're not world ending, as opposed to things like Alzheimer's or diabetes. These can be really awful things that can potentially be or are caused by viruses. So Ebola, guys, is going to be a condition where the virus is going to cause such massive bleeding on the inside of your body that you bleed out of every orifice, and I mean every single orifice, every hole you have. You're going to bleed out of it because the blood needs to find a way out, and that is how Ebola works. So bleeding from every hole. That is a really bad virus. Before we had a vaccine for it, rabies was a death sentence. So think of it like that. Again, you don't need to know examples. These are just ways to think about all of the variety of viruses and how it affects humans. So with all these viruses, how the heck do we fight them? What do we do to treat a viral infection? The short answer, nothing. We can't do anything. Once you are infected with a virus, you can treat the symptoms, and that's a little bit it. When it comes to having the flu, there is an antiviral drug, but you have to take it within the first 24 hours of being infected, and most people don't know that they have the flu until they start showing symptoms, and once you start showing symptoms, it's usually after 24 hours, so the antiviral is kind of pointless. Because you didn't even know you were exposed to it until you start having a sniffly nose, having a cough, sore throat, etc., fever, the whole thing. So in having to tell somebody that, hey, you have this virus, I'm sorry, you just got to ride it out. There are no quick fixes to a viral infection. You just got to treat the symptoms and try to make yourself comfortable, because antibiotics will not work. An antibiotic is going to treat a bacterial infection. A viral infection is a virus. Antibiotics do not work on a virus. Very important thing to make a note of. The one thing that we do have to kind of fight viruses is prevention, and that is through vaccines. However, there are many reasons why vaccines kind of fail, and especially more recently, it has a lot to do with kind of a fear or misunderstanding or a mistrust of vaccines for a bevy of reasons that we will eventually talk about in a little bit more detail. But there's really kind of a need to either A, fix the image of vaccines so that they can start being used more effectively again, so that we start having an immunity to viruses and we don't start seeing things like polio pop back up, or develop a better fix for viruses that have less of a fear and mistrust surrounding that treatment. And with that, my friends, that is the end of Unit 1. Everything up to Chapter 5 is fair game for Exam 1. Make sure to be studying. There is a lot of information that you need to make sure that you know for Exam 1. If you have any questions ever at all about anything, please make sure you email me, message me in any capacity. Make sure to be studying. Watching all the videos over and over would probably be my best recommendation to study for this first exam. With that, guys, happy studying, best of luck, and make good choices.